Zooplankton, the animal drifters in the ocean, are important food for birds, fish, and marine mammals, so understanding how their abundance and distribution changes is key to understanding our ocean ecosystems. This summer, I worked on validating a molecular zooplankton detection system called SHA, Sandwich Hybridization Assay, and it basically sandwiches ribosomal RNA between two probes and gives us a signal that is a proxy for organismal abundance. First, I had a known signal that I got from copepods, which are a type of zooplankton that were grown in the lab, and then I added those copepods to 2 liters of water taken from locations around Monterey Bay to see if the signal would be consistent. It was difficult to do this because my water samples contained organisms that were picked up by my test. However, I was able to use algal lab cultures to show that we might expect our test to perform consistently even with bloom concentrations of a toxic phytoplankton called Pseudonychia. This is important because we might want to know how the toxin propagates up the food web into organisms that we might eat, such as fish. Lastly, I showed that things like size, physiology, and life history stage of an individual can influence our results. For instance, tiny newly hatched copepods that are about the width of a human hair gave us a larger than an expected signal, perhaps because they have more RNA since they're actively growing at that stage. Overall, sandwich hybridization assay is a robust method that we can use to detect relative abundances of organisms, and my project helped understand what our test signal might indicate about zooplankton ecology.